Okay, discovering drugs. Traditionally, drugs are extracted from plants or microorganisms such as moulds, and you can see I've colour coordinated the ones that are from plants and the ones that are from mould. So two plant examples first. So foxglove, they're like a purple flower, has got digoxin in it, and that is used to treat heart problems. It sort of strengthens the heartbeat and that sort of thing. It's still used nowadays. There are other drugs that come from this plant as well, but this is the main one. The second one is willow tree bark. So willow trees, you often find them by rivers, and they've got long leaves and branches. There is a compound used in the bark of this tree that makes aspirin, and actually this has been used as far back as 400 BC for things like pain relief. So moving on to penicillin, many of us will know this is an antibiotic. It was discovered in the early 20th century by a scientist called Alexander Fleming. Now he discovered it by accident, but this is one of the first antibiotics to be created. So just to go through the process of how he found it out. So Alexander Fleming was growing bacteria for study purposes. He was quite careless and he left the lids off his petri dishes. So they're like little round dishes that got jelly in them, and you can put a lid on them, but he left the lid off. This means that bacteria and other things from the air could go and land on these dishes. So he went away on holiday for a bit, and when he came back, mould had started growing on his culture plates on these dishes. He noticed, however, that there was a clear ring of jelly around some of the mould spots, and this meant that the bacteria had been killed around the mould. So the bacteria had been killed by the mould that had grown. So Fleming recognised the importance of his observations and named the substance that killed the bacteria penicillin. He tried unsuccessfully for several years to extract the active juice from the mould, but he gave up because he just couldn't do it. However, about 10 years later, two other scientists called Ernest Chain and Howard Florey set about extracting the penicillin, and they actually succeeded at doing this. Chain and Florey gave penicillin to a dying man who had a blood infection, and he almost recovered, but then the penicillin ran out. So unfortunately that man died, but until they stopped giving the penicillin, it was actually curing his bacterial infection in the blood, and this gave Chain and Florey evidence they needed that penicillin killed bacterial infections. Florey and Chain then started working on with the medicinal company Pfizer. These still about nowadays. They're an American company. They started to produce this on an industrial scale. This saved many lives, and they're still doing so today. Looking at medicines for the future. Now, most new drugs are synthesized by chemists in the pharmaceutical industry. So synthesized means that they are made artificially. So we haven't found them naturally. However, having said this, a starting point for many new drugs is a chemical extracted from plants. So we can get a plant, it knows it has certain properties, we know this chemical has certain properties, and we can copy the molecular structure of it and then synthesize that. Compounds showing promise as antibiotics can be modified to produce a more powerful molecule that can easily be synthesized and done cheaply. So we can test lots of different things. If something's shown promise, or a certain property in that chemical is working how we want it to, we can modify, add things in and out, and make it more powerful. And finally, discovering new antibiotics is very hard. Scientists are using microorganisms found in the soil to do this. However, only about 1% of the microorganisms in the soil can be grown. So it's quite hard to actually use the soil, because 99% of it we can't actually grow. We can't actually keep it alive to actually test it. But they have found a way of doing this, and in 2015 they actually discovered a new antibiotic that kills all bacteria, even the resistant bacteria we've got now. It's still in its trialling phase, but this is very promising for sort of curing things in the future, so that we don't have a problem with antibiotic resistance.